and we're going to talk about genetics and genetic disorders. So, first question, what do we mean by genetics? Don't all answer at once now. Things that are in your genes? Okay, and what is a gene? I heard someone say DNA. What's the relationship of DNA to gene, or gene to DNA? Close. I mean, yes, they do both make proteins, but it's more specific. Okay, so a gene is a portion of DNA that codes for a specific protein. Does that make sense? So a gene is a piece of DNA. And where does DNA live in human cells? In the nucleus. And what's the purpose of DNA? To make proteins. So there's a bunch of control mechanisms that are also contained in the DNA, but they all boil down to making the protein or not making the protein. So to simplify things, what is the purpose of DNA? To make proteins. And it's those proteins that make up us as humans. Name 5 proteins that are in your body. Albumin, all right? Give me another one.
Sorry, saying it, but it was like...
Yeah, I wasn't sure if that was actually one or not. So albumin, give me another one. What carries oxygen? Hemoglobin. Give me another one. Makes your skin waterproof. Your hair is made out of it too.
Keratin.
Keratin. All right, makes your skin strong and also scar tissue.
Collagen.
Collagen. One more. Okay, elastin is what makes your skin stretchy. All right, so you got 5 proteins right there. All of those proteins are coded for by DNA. What about the protein that makes your skin darker colored? Melanin. Okay, so that's another one, and so that's another protein that is coded by DNA. Now, the skin color one is good example because not only does it code for it at all, but it also codes for how much of it you produce. Which is why some of us, when we go out in the sun, we just go straight to red. You know, white, red, burn. Others get this nice, beautiful tan, and we're like, man, wish I could tan like that. I just burn. But, so all of that is genetically coded. Not just does the protein exist, but how much of it gets expressed and under what circumstances. So genetics helps to control all of those things. Does that make sense? Okay. So in our bodies, in our human bodies, as opposed to our animal spirits, how many chromosomes? What is a chromosome?
It's like balled up DNA.
Okay, so it's balled up DNA. It's a bundle of DNA. All right, so how many chromosomes, how many little bundles do we have inside of our cells? 23 pairs. Are you trying to make me do math?
Well, 46 total.
Okay, so it's 46. And why do we call them pairs? Why do we group them into pairs? Because we got half of them from our mothers, and we got half of them from our fathers. And in this case, we don't necessarily mean the people who raised us. We mean the person who donated the sperm and the egg, whether that was a natural process or a not so natural process. You're looking at me like, yes, they're...
I did not look at you.
Yeah. Yeah. So most of us were born through the natural method, although some of us may not have been. And in the future, who knows? Maybe we'll all end up like Gattaca. Have you guys ever seen the movie Gattaca? You should watch it.
What time?
Tonight. How's that? Maybe I'll put a bonus question on your test about Gattaca.
That's not.
So
Take the time and study.
it is studying.
For one
It's about genetics. So it's about some of the dangers inherent in genetic testing, but we'll get to that later. So chromosomes, there's 23 pairs. one of each pair comes from our mother, one of each pair comes from our father. If you're missing if you're missing a chromosome, or if you have an extra chromosome, that can lead to a certain number of diseases. The most famous of those is Down syndrome, and that's going to be caused by an extra chromosome 21. And so there's proteins that are coded on that protein 21 that when you have too many of them leads to Down syndrome. So if we take all of your chromosomes and we line them up together in pairs, what do we call that little graph? It's called a karyotype. So if you're looking for genetic disorders, one of the things that would be done is to do a karyotype. If you see an extra chromosome, well, there's maybe not the problem, but a problem. If you're missing the chromosome, well, there's a problem. Now, in order for chromosomes to be functional, for DNA to work, what has to happen to the DNA? Okay, so replicate. Replicate into what? We got to make a copy of it, right?
Yeah.
Okay. So, can you make a copy of something that's bound up together?
No.
What do we have to do? We have to unwind it. So, the name for the unwound DNA that's ready to be used is chromatin. So, chromosomes are kind of wound up, and they're wound up for the purpose of replicating. So, mitosis. So, when the cell mitoses, you don't want to be dragging something that's like a mile long. Come on, get over here. So what your body does is it bundles it up into a chromosome. It's kind of like a ball of yarn. But in order to use the chromosome, the DNA inside the chromosome, we've got to unwind it. And that's called chromatin. So what is the structure of DNA real quick? A double helix. What does that mean? All right, so you've got two strands, and they wind around each other. So if we take this and we expand it, you're going to see something that looks kind of like this, right? And there's going to be a matched something on each side, right? Okay, so how many base pairs are there? How many bases are there? How many possibilities? 4. So what are the 4 bases? Do you remember? ATCG? Adenine, cytosine, guanine, and thymine. So A matches with T, and C matches with G, and the other way around. So one of these is the one that's like the master copy, and the other one is just a mirror image, right? So in order to make this work, we have to unwind it, then we have to unzip it, and then we've got to match up. So we're going to get rid of this copy just for a moment. And so we're going to match up our C's and G's and A's. So let's say that we have this A, T, G, C. What are we going to get matched up over on this side? Are you sure about that?
Yeah.
What kind of copy are we making? To make a protein, what do we have to do? We have to make RNA, and in RNA there's no T. It's you. It is me. No. Okay, so U, and then A, and then C, and then G. So this is going to be our DNA original, and this is our messenger RNA copy. Does that make sense? So after we make this copy, the DNA rezips back up together. Does that make sense? So what happens is you're unzipping, making a copy, and then rezipping right behind it, if that makes any sense at all. And if it doesn't, whatever. So what is this process of making messenger RNA called? Well, it's part of protein synthesis. Transcription. So when we transcribe, we're just making a copy. Now, what kind of copy is it? It's a mirror image copy, right? Okay. So now that we've got this little strand of messenger RNA, what happens to it? Okay, what is translation? I don't know. I just memorized it in Bio 1, okay? It was like 2 years ago. Okay, we're going to make a protein, but where do the proteins get made? In the ribosomes. Now, this is going to be mind-blowing. What is RNA? Ribonucleic acid, because it's got ribose in it. Now, where does RNA work? In the ribosomes. Ah. Never thought of that before, did you? So, our little messenger RNA is going to leave the nucleus and go to a ribosome. In the ribosome, what we're going to do is we're going to match up each of the codons. What's a codon, by the way? Groups of 3. I'm drawing them with visible space between them just to emphasize that we're grouping them into 3, but in real life it's not that way. So let's just say we had A, T, so not A, U, C, 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 G, U, G, C. I do not know if these are actual codons. I'm just making them up, so don't, just example. So, how many codons do we have in this example? We have 3. So how many amino acids are we going to code for in this little example? 3. Each codon codes for 1 amino acid. Okay, so what we're going to do is we are going to match this up. So what's the match for AUC? UAG. And so this is connected to something called transfer RNA. And what's bound to this transfer RNA? An amino acid. So now we're going to do the same thing with the next one. CCG matches to GGC. And there's a different amino acid here. So each transfer RNA associates with a different amino acid. And then the ribosome allows these 2 things to be matched together, and then what does it do between them? A bond. What's that bond called? Peptide bond. So in the ribosome, we're going to match up the messenger RNA to transfer RNA. Transfer RNA consists of how many bases? 3. So we call those 3 that it matches to a codon. So we match the codon to the transfer RNA. The transfer RNA is associated with the specific kind of what? Amino acid. So then once we match 2 codons that are next to each other, what do? We make a bond. What kind of bond? Peptide. So as this runs through the ribosome, what we're getting out the other end is a chain of amino acids that are bound together by peptide bonds. And what is the name for that? A polypeptide. Okay. So is that all that we have to do to make a protein? Or is there more? No, there's more. So polypeptides don't stay in a straight line. What do they like to do? How many of you ever worked with saran wrap? What does saran wrap like to do? Stick to itself, right? So and you're like, you can never get it flat again. Okay. So these like to stick to themselves, and so they'll often wind themselves up into shapes. So here's one simple example where you've got this, I think I did it upside down, but you get the idea. So the polypeptide chain winds itself around, and then you get these extra little bonds called sulfide bonds. So here we have a protein, right? But it might not be a usable protein. So in order to make it into its final usable form, we have to use enzymes to cut it. So let's say we cut it there and there. And now we end up with a protein that looks like this and a protein that looks like that. So this right here is insulin. And what's this right here? It's a piece of trash. But What happens if you take it that way and you turn it this way? What does it look like? A C. So we call this little piece of trash protein C peptide. So in order to make a protein, first we start with, what's this? We make a copy. What's that called? Transcription. Then we have to turn that messenger RNA. We have to match it up with tRNA and bind amino acids together into a peptide, polypeptide. The peptide bond then rolls in on itself and you form sulfide bonds. And then the last step is to cut it with enzymes. Now, in biology, this leads to kind of like a chicken and egg problem. DNA codes for proteins, but it doesn't work unless there's already proteins present, right? What are ribosomes made of? Proteins. What are the enzymes that do the cutting? Proteins. So it's like, on the one hand, DNA makes the proteins. But on the other hand, if you don't already have proteins that exist to do the work for the DNA, it can't do its job of making proteins. Does that make sense? Okay. So, which is one of the reasons why it's so hard to make life out of nothing. But this process is kind of the basis for genetics. Now, this is happening in every cell in your body that has a nucleus. Tell me a cell that doesn't have a nucleus. I mean, a cell in your body.
Oh, sorry.
Yeah, red blood cells. Okay, give me another one. It's not necessarily a whole cell, it's a part of a cell. A cell fragment. Looks like you could eat off it.
Oh.
Platelet? Sounds like. Okay, so platelets. So both of those things can't produce new proteins because they're missing DNA. But all the other cells in your body that have nuclei, they can produce proteins through this process. Now, what is it that makes a skin cell different from, say, a lung cell or a muscle cell? You got nothing. You had no idea. Is it the number of genes? Do they have different chromosomes? Or do they have the same chromosomes? They have the same chromosomes. Well, if they have the same genetic material inside them, how come one makes melanin or collagen, and how come the other one makes myoglobin? How come one makes elastin for your skin, and another one is making hemoglobin for your blood? I just don't understand. What? Say again?
They code different proteins.
They code different proteins. Oh. So there must be some way that we can turn certain genes on and certain genes off. Right? Because otherwise every cell in your body would make all of the same proteins and you'd have no tissues. Nothing would be differentiated. It'd all just be one globby mass. So in order to make you into an organized organism, so organism is organized, get it? So in order to make you into an organism that's organized and works, some cells turn on certain genes and they turn off other genes. Does that make sense? Okay. So what is the whole idea behind stem cells? What can a stem cell do that other cells can't? They can become different kinds of cells. So as a cell goes from having lots of potential into I'm dedicated now to being a skin cell, what is that process called? What? Differentiation. So the more differentiated it is, the more different it is from all the other cells. The more different it is from that immature cell that can become anything. So think about it, psychologically speaking, what could a little baby become? It could become an astronaut. It could become a nurse. It could become a firefighter. It could become a supervillain. That little baby could become anything it wants to be, right? Well, kind of. But eventually, as it grows up, it's going to become more one thing than other things, right? That's why what's interesting is like, listen to high schoolers talk about what they're going to do. I'm going to be a supermodel and a doctor. I'm going to be a firefighter and a policeman and a model. Recently there's a girl who was doing CPR and a guy saved his life in Chick-fil-A. And so, what she's going to do is she's going to be a firefighter and a doctor. And so, why do high schoolers phrase things like that? Because they're naive. But more important, they're not that differentiated. So they've got potential, and I could do this, and I could do that. But as you get older, you realize you can't do all that. And it's going to be more this. And so I'm going to dedicate myself to this. And so that's what happens to cells that are no longer stem cells as they become dedicated to a particular thing. So one of the things that we want to try and do when we do stem cell type therapies is we want to identify cells that have the potential to either go back to being a stem cell, or we want to take cells that are closer to being that stem cell already. So if you take a look at the stem cells in your blood, those have the potential to become red blood cells, white blood cells, and within white blood cells, you've got like 5 different types of white blood cells. So they already have the potential to become more than one kind of cell. Does that make sense? Mm-hmm. And so that's what we're talking about when we talk about stem cell therapies, is we're taking stem cells that have the potential to become different things, putting them into a person and giving them hormones that generate them, or kind of influence them to generate in a certain direction. Now, we'll talk a little bit more about that in the future. But the important thing is, out of this whole thing, is that each cell has some genes turned on, other genes turned off. And so there has to be control mechanisms for that. And we'll talk about what some of those control mechanisms are as we get a little bit farther along. Now, at this point, what we need to do is we need to talk about inheritance patterns. So, how many of you remember Punnett squares? Me, I remember Punnett squares. So, there are a number of disorders or diseases that are caused by a single gene mutation. So, if that single gene is not present on either the X or the Y chromosome, what are the other 22 pairs of chromosomes called, by the way?
Autosomal.
Autosomal. And the pair that is not autosomal are called sex chromosomes. So X and Y are sex chromosomes. Everything else is autosomal. So there's a number of autosomal genes that if you have one single problem can create a disease. So we separate those two things into either autosomal dominant or autosomal what? Recessive. Now, these terms came about by a guy named Mendel. He's a monk back in, I guess, 1800s. And he was just observing. And if you remember, he was observing, I think it was pea plants, right? So he was observing the effects. So what he was saying is these dominant traits tend to dominate when they're around, and these recessive traits tend to only show up when it's the only thing that's around. But that's not really the right model to think of. It's not that one is strong and the other one is weak. The actual problem is that, or issue, is that dominant is a protein present causes the disease. Yes, Huntington's disease is an example of that, of dominant. In recessive, it's a missing protein causes the disease. So if you have one gene that's producing, gene. So the gene codes for a specific protein, right? Right? Now, if there's one single gene, how many copies of that gene do you have? I just told you.
1, 2, 2.
Two. One from your mom and one from your dad. I guess mom is left hand, dad is right hand today. So one from mom, one from dad. And so you've got two potential versions of that gene. What are those versions called? Starts with an A. It has too many L's, allele. So you've got two alleles, two potential expressions of that gene. So out of those two alleles, so we'll call one mom and one father. Mother, father. Okay, so we have a mother and a father allele. So it doesn't matter which of these it is because they're interchangeable. But if you have one of these alleles producing that protein that causes the disease, you're going to end up with the disease eventually. Does that make sense? So in the case of Huntington's disease, you're producing a protein that accumulates in the brain and eventually poisons the nervous tissue. And that's why you don't get the disease until later in life. But you're producing the protein from the moment you were born. Does that make sense? So we don't need to fix the gene in order to fix the problem. I mean, we don't need to replace the gene. All we have to do, what do we have to do to fix the problem? Turn it off. We hadn't flipped the switch on that gene. So if we can either flip the switch, turn it off, or if we could just dial it down so it's not producing so much, either of those would effectively treat or prevent Huntington's disease. Does that make sense? Okay, so there's a number of these disorders. Another one that you may have heard of is called Marfan syndrome, which is a genetic disorder related to collagen, where you produce a collagen that isn't as strong as it should be. So patients with Marfan syndrome get injured more easily, and they have a harder time healing than normal people do. So those are two examples of autosomal dominant disorders. So what we have to do is understand the genetic pattern of them. So I like to use pluses and minuses instead of big H and little h. I just think that's, if the whole idea is there is a protein or not, well, if there's a protein, it's plus, and if there's not a protein, it's minus. Does that make sense? So let's imagine for a moment that everyone is normal. So your mom is completely normal, and your dad is completely normal. So mothers will be on top, fathers will be on the side. What is the only choice your mother can give? Normal. So your mom is always going to be giving normals. What's your dad going to be giving? Normal. So if both your parents are normal, what does the kid have to be? Normal. Now let's imagine for a moment that mother has Huntington's disease. So she's got two possibilities. She can give positive, so she's positive for the protein, or she can give negative, and she's negative for the protein. So her kids, half of them will be positive for Huntington's, and half of them will be normal.
So each kid gets isn't one block.
Yes. Yeah. So you get each block represents one possibility of a child. So on average, yes. So on average, half the kids will be Huntington's and half the kids will be normal. Now, what's the key word there? On average. But you can have incredibly lucky genetics, and this couple could have 6 kids and none of them have Huntington's. Or you could have 6 kids and all of them have Huntington's. There's no guarantee that this next child will have it or not. It could go either way. Does that make sense? So that's the idea behind Huntington's disease and other autosomal dominant. What was the other autosomal dominant disease that we mentioned? Marfan syndrome. So there's a few others, but those are the only two we're going to mention in this course. The key pattern is that there's no such thing as a carrier. You either have it or you don't. Or as Yoda would say, either do or do not. There is no carrier.
Is that just for autosomal dominant?
Yes,
Oh, okay.
that's for autosomal dominant. There's no carrier. You either have it or you don't. And what are the chances? What are the chances that if one of your parents has a disease, you'll have the disease? 50-50. Does it matter for boys or girls? No, because it's autosomal, not sex-linked. All right, that one is pretty simple. There's only one scenario.
Don't families then know that they have it?
You know that it runs in your family, yes. So, all right, we're going to talk about this later, but we'll talk about it now. Can we genetically test for this? Yes. Now, how many of you think that if your mom or dad had Huntington's disease, that you would like to know whether you have it or not? I think I actually talked about this last time.
Yeah.
Yeah, but as it turns out, most people don't. And the rate of suicide among people who find out that they're positive for the gene is very high.
What if dad has it too?
Okay, in rare cases, you could have both a mother and a father who have it. So in this case, you could get a positive from your father. So in this case, you got positive, positive, negative, negative, and then you've got mixed. So these two are going to have normal Huntington's disease. This person is going to be producing twice as much of the protein as usual. So they'll have much more aggressive Huntington's disease. And then this person would be normal. So in this case, there's a 1 1/4 chance any given child will be normal. There's a 3/4 chance that they'll have Huntington's. And then there's 1/4 out of all of them that'll be very severe
And
Huntington's.
that very severe, well, they probably wouldn't have children, or maybe they would, but their children would definitely have it because they're like positive.
Yeah, if this person has children, they're going to have Huntington's because there's nothing they can give that's normal.
What if they got, like, their grandparents had Huntington's, but they had, like, both negatives, they're normal? Could their children eventually get it?
Okay, it can't skip a generation in that way.
Okay.
The way it could skip a generation is if so let's just do that for a second. So your grandparents, one of your grandparents had it. Your father does not have it. Let's say it was your father's grandfather. Your father's father, your grandfather. He's got Huntington's, but your dad doesn't. There's no way that then he can somehow magically give it to you.
I feel like there's a slight possibility that you could just have it in your genes, but just doesn't show.
No.
No,
Okay.
if you're positive, it shows.
It should show. Now remember, well, we haven't talked about this yet. So there's variable expressivity. So there is a possibility that it could skip a generation if some reason the gene turned off in your father, but he gave it to you and it's turned back on.
Like he had the positive, but it was turned on.
Yeah. So in that sense, it's possible, but with Huntington's disease, it's not very likely. The other thing that I should also mention is that when we're talking about the gene as though it's an all or nothing, right? But this positive is not just one single gene. There's at least a dozen different varieties of Huntington's genes. So some of those genes code for just a little bit of protein. You get the disease later in life. Some of them code for an enormous amount of protein. You get the disease much earlier in life and more severe. Does that make sense? So just because you're yes or no doesn't tell you how severe the gene itself is. But then we also have to take into account how expressed that gene was. What do we mean by expressed?
Is it like the two positives and the...
No. Okay. How many of you have ever lived in a house that has a dimmer switch?
Oh, yeah.
The lights are on or off, but they can be brighter or dimmer. So when I say variable expressivity, just because the lights are on doesn't mean they're really bright. So just because a person has the Huntington's protein doesn't mean they have an enormous amount of it. The smaller the amount, the later the onset and the less severe the symptoms.
So they can potentially go their whole life without really knowing that they have it.
Potentially, especially if they die when they're relatively young.
Okay.
If they have a heart attack at 50,
Yeah.
they may never have lived long enough to experience it. So it skipped, but really they just died too early to experience it.
When they, I forgot what it's called, when you die, could they test to see that you?
Autopsy? Well, you can test right now.
Yeah,
Yeah.
no, but
Are
you.
you saying test the brain?
Yeah.
Yes.
Is this variable expressivity
Variable expressivity.
only for autosomal dominant or for all of these?
It's for everything. Well, sorry, lies. It's only for things that are there. In the case of autosomal recessive, it's something missing. So you can't make something that's not there. Whereas you can make less of something that is there.
Okay.
Okay. So autosomal dominant, you guys went down a giant rabbit hole that's not going to be on your test.
Oh.
You kept asking questions. I was just answering your hypotheticals. The important thing is this inheritance pattern. Half the kids be normal, half the kids will have the disease. No such thing as a carrier. Can you remember those three things?
Yeah.
All right. So autosomal recessive now, we're missing a protein that needs to be there. Or the protein that we're making is faulty and doesn't work correctly, which is the same thing as saying you're missing the functional protein. So the classic example here is cystic fibrosis. And in cystic fibrosis, you're missing the gene that correctly codes for chloride channels or chloride gates. And as a result, you have a hard time moving chloride and water where you need it to go, because sodium and chloride tend to go together. And if you can't move the chloride, you can't move water as well either. The hallmark of cystic fibrosis, originally, before we understood what it was that when you kissed your child's forehead, it would be salty. So, you know, like when you sweat, there's like a little bit of salt produced. Well, and their heads would have so much salt that it would be really salty. And that was how you knew that your child was not going to live very long, because cystic fibrosis kids usually died before age 2. Now, with modern medicine and treatments, we can keep them alive till the average lifespan now is around age 50. So we went from age 2 to age 50. That's pretty good, all things considered. So you're missing that gene that makes that protein that creates the chloride gates. So there's a couple possibilities now, because we're talking about something that's missing. So in this case, we're going to have mom on the top, we're going to have father on the side, and in this case, we're going to make the father have the gene. So in this case, positive means you've got what you'll need. Negative means you're missing what you need. So mom is normal and dad is a carrier. Sorry, mom is a carrier, dad is normal. It's so confusing there. So mom is a carrier because she's got one missing gene. Dad's got 2 normal genes, or 2 normal alleles. So dad is always going to give a positive to all the children. And then mom is going to give on this one positive, and over on this one negative. So if you've got at least some of the chloride gate, you're not going to have cystic fibrosis. So how many of these children will have cystic fibrosis? None. So if you have a normal parent and a carrier parent, all of the children will be normal in the sense that they will not have the disease. But genetically speaking, what do you have? Half of them, 2 out of 4, half, are carriers. So a carrier marrying a normal person, how many of them will have the disease? None. How many will be carriers? Half. Does it matter boys versus girls? No.
So if I have a friend that has the cystic fibrosis, but neither her parents do, they both were carriers?
Yes. We'll get to that in a moment. Okay. So the important thing to remember is if you have a carrier marrying a normal person, none of the children will be cystic fibrosis themselves, but half of them, on average, will be carriers. Now, what happens when you get two carriers who marry each other? Okay, so in this case, the father is going to give his normal allele to half the kids, and he's going to give his abnormal allele to the other half. Mom is going to give her normal allele to half the kids, and then her abnormal allele to half the kids. So if you have any positive, you will not have cystic fibrosis. So how many kids out of these 4 possibilities have cystic fibrosis? 1. So 1 out of 4 means 25%. So a quarter, 25% of the children will be cystic fibrosis. Out of the 3/4 that are not, out of the 3 possibilities that are not, 1 will be completely normal, and the other 2 will be carriers. So what are the chances that a child could be a carrier? 50%, 2 out of 4, which is the same as saying 1 out of 2, which is saying 50%. Man, fractions are so hard.
Wouldn't it be 3 out of 4 because technically the one with it? Oh, they're not, but they carry.
This is not a carrier, they have the disease.
Carrier.
Yeah.
And they can pass it on.
Yes, they can pass it on, but they are not a carrier.
Okay.
A carrier means you don't have the disease yourself.
Okay.
So, all right, so now,
Does this go for sickle cell and PKU too, or is it different?
it's the same for all of the autosomal recessives. So PKU and sickle cell and the others that we're not discussing in this course. Now, let's say we take that child who's got cystic fibrosis, and through the miracle of modern medicine, that person lives long enough to have children themselves. So now, let's say it's the dad. The dad has cystic fibrosis, so he has no choice but to pass it along. So we're going to end up with dad giving his cystic fibrosis gene, and then mom has no choice but to give normal allele. So how many of the children will have cystic fibrosis? None. How many will be carriers? All of them. Does that make sense? Okay. So on the test, it's going to give you a disease name. It's not going to tell you this is autosomal, recessive, dominant, whatever. You're expected to know, oh, this type of disease is this type of pattern. Then it'll give you a scenario. You know, this kind of mother marries this kind of father. What do you tell them about? And the questions could be like, how many of the children will have the disease? How many of the children will not have the disease? How many of the children will be carriers? Yeah, and so you've got to figure out in that specific situation what to tell those
So like
parents.
a cystic fibrosis carrier marries a normal person.
Right.
Near the kid. Okay.
Yes. So for those of you who didn't hear that, so you've got a cystic fibrosis mother who's marrying a normal father. What are the chances that their kids will have cystic fibrosis?
But it'll say carrier or
Yeah,
disease. Right?
it'll tell you what the parents are.
Oh, okay.
Yeah. All right, any questions about cystic fibrosis in particular? Okay, so let's just briefly discuss sickle cell and PKU. So PKU, phenylketonuria, is a disease where the enzyme responsible for helping to break down phenylalanine is missing. So phenylalanine is a amino acid. It's one of the amino acids present in aspartame. Is that right? Yeah, who cares? I may edit that out. I may not. Phenylalanine is an amino acid. It's present in a lot of foods. And if you look on food packages, you'll see that a lot of times it'll say, this food contains phenylalanine. And what that means is, if a person with PKU eats that food, they are not going to be able to process the phenylalanine correctly, and it'll end up in this byproduct called phenylketones. Now, in adults, it can cause some problems, but it's not life-threatening, usually. But in little children, it can poison their brain so they become permanently brain damaged. So the very, very important thing with PKU children is that they should never get phenylalanine foods until their brains are fully developed. And even then, you really should try and avoid phenylalanine. In the United States, we test pretty much all kids for PKU at birth, which is kind of silly because if it doesn't run in your family, well, the chances that you're going to develop it spontaneously are about as tiny as your little head that I'm crushing right now. So it's an enormous cost to have that screening program, and how many children does it actually catch? No, it's not that common. So that's PKU. The next one is sickle cell anemia, and sickle cell anemia is caused by a mutation in the hemoglobin gene. So instead of having normal hemoglobin, which is called hemoglobin A. Do you remember what hemoglobin A stands for from Pathopharm 1? Well, what's the opposite of a child? An adult. Right. So the major types of hemoglobin A is adult, hemoglobin F is for fetal, and then hemoglobin S for sickle. So when hemoglobin S, sickle cell anemia type hemoglobin, is exposed to cold, it changes configuration, and it makes the red blood cells go from being flexible to being kind of like a hard sickle shape, which is kind of like a C. And as a result, they start to break, and they also can't fit through capillary beds. So to fit through a capillary bed is actually smaller than a red blood cell. So the red blood cells have to kind of bend in half, which helps them to release their oxygen. But when it's that hard sickle shape, it can't do it. So it builds up at your capillary beds and causes sludging, which causes pain in those patients.
And is it harder to breathe?
It is harder to breathe, probably. But the key thing there is that on the one hand, they're destroying red blood cells. What kind of anemia would that be called? When you destroy red blood cells? Nope, that's when you're not producing them. Hold on, let me about it. What could it be? What's it called when you break a red blood cell? What's the fancy name for blood? Hemolytic anemia. Right, I knew that all along. I was just joshing you, Dr. Heyman. Yeah. So hemolytic anemia. So the patient's going to have lower red blood cell counts. So it's going to make it harder for them to breathe in the sense that they'll be more short of breath and they'll have all those classic signs and symptoms. But at the same time, because they're having that sludging in their capillary beds, and it's actually causing damage to the capillary beds, they're going to have pain in their extremities because you're actually damaging the blood vessels. And what's the body's response to damage? Inflammation. So they're going to have inflammation in their extremities where those red blood cells are sludging up. So that's basically what happens in sickle cell anemia. Now, as long as you have some normal adult hemoglobin, you don't end up getting sickle cell anemia. It's only when you don't have any adult hemoglobin, you only have the sickle cell hemoglobin, that's when you get the problem. All right.
So what if did you get a buildup of the sickle cell anemias, and then you get just a buildup in your arteries and veins, or no? Is that not a thing?
Well, yeah, it's a thing.
And then it could pop, and then it could go to along with that sort of situation.
Okay, so it typically doesn't pop through.
Yeah.
It just sludges up against the capillary bed.
Okay.
So, but yeah, you guys will learn more about sickle cell anemia later on. So right now we're just introducing the genetics of it primarily and then giving you just a brief overview of the disease. All right, But those patients, in addition to treating their anemia, they also need to be treated for pain. Now, these are autosomal, which means that it's not in either the X or the Y. But we do have some diseases that are recessive, but they're on the X chromosome. So we will call these X-linked. The two classic examples here are color blindness and hemophilia. There's multiple types of hemophilia. I can't remember which one is this. Doesn't matter. Hemophilia. So in the case of color blindness, we're missing one of the proteins that colors for, or codes for the color distinguishing cells and components of your retina. And in the case of hemophilia, we're missing one of the genes that codes for clotting factors. Now, the key difference with X-linked is that it matters mother versus father. Why is that? Because a mom is always going to be an X. And a dad is always going to be an XY. Okay. So let's imagine for a moment that we have a father who's got color blindness. So he's missing, I guess we should put it at the top. He's missing the color producing proteins in his eye. Or at least he's got faulty ones. So he can still see kind of colors, he just doesn't see the right colors. So in this case, he has no option. He's always got to give his faulty gene. Does that make sense? Or his faulty allele. And then on the Y side, he always gives his Y. Now the mom, let's say that she's normal. So she's always going to give normal. Okay. So in this case, in order to be colorblind, you have to have no normal X's. Are there any children who do not have at least one normal X? No. So how many of your children in this case will be colorblind?
None.
None. Now, how many will be carriers? 50%? Which 50%? The top 50. Now, how is the top different from the bottom? Girls, boys. So 50% of the children will be carriers, but how many of the girls?
All of them.
All the girls will be carriers. What about the boys? None of the boys. The boys will be completely normal. So a father cannot pass on an X-linked disease to his sons, but all of his daughters will be carriers, assuming the mother's normal. Does that make sense? All right, so now what we're going to do is we're going to have one of those carrier mothers marry a normal guy. All right, so the dad always gives his normal X or his Y. And then the mom, half the time, is going to give her normal X. And then half the time, her missing allele. All right, so in this case, let's look at the girls by themselves first. What will the girls be? Half carriers and half completely normal. How many of them will have color blindness? None. Now let's look at the boys. So half the boys will be normal and half the boys will have color blindness. So where does a man get his X-linked disease? From his mother. So you can inherit it from a mother who was a carrier who got it from her mother, or you can get it from a mother who was a carrier who inherited it from her father. So the carrier can get it from either a father or a mother. But only mothers can pass it to their children. Fathers can never pass it to their sons. Does that make sense? Okay. So that's X-linked. And what are the two major diseases that are X-linked? Hemophilia and color blindness. So same thing on the test. You'll be given a scenario. You've got this kind of father. You've got this kind of mother. What are the chances that blah, blah, blah. Okay. All right. Any questions so far about any of these disease inheritances?
So on the test, we should know the disease that for each type.
Yes.
What was the second disease for autosomal?
Marfan syndrome. Marfan. All right. So at this point, we're going to talk about something called polygenic inheritance. So everything we've talked about up till now is controlled by how many genes? One single gene. So how many possibilities are there? Two. Okay, so let's talk about eye color just for a moment. So eye color ranges from very light blue all the way through very dark brown, right? And what is it that controls the colors in between? What is it that controls those color variations? Yes, genes, but I'm talking about physically. What does a dark-eyed person have more of that a lighter-colored eyed person does not? Melanin. So it's the same stuff that makes your skin dark, but it's in your eyes, in the iris of your eyes. As it turns out, there are 4 genes that control eye color. So how many possibilities does each gene have? 2. So how many total possibilities are there? 8. Okay, so if you have...
Wait, 4?
There's 4 genes, but there's 2 alleles per gene. So let's just say that this one is from your mother, this one is from your father. Now, if none of them are positive, you can have very, very little melanin in your eye, and you have very light blue eyes. If all of them are positive, you're going to end up with very, very dark brown eyes. Does that make sense? Mm-hmm. Okay. And if they're in the middle, you have hazel or green eyes. So what happens when you have parents? So let's say you have parents, the mother has got all positives. What is the only thing she can give? She can only give positives. Now, if the father is the same, what's the kid going to be? Brown, dark brown. It's like, that's pretty simple. That's easy, right? No?
No, it can never be that easy.
Well, actually it is. There are complications, but 99% of the time, it's that easy. If both the parents have very dark brown eyes, then the chances that the kid will have dark brown eyes pretty much that.
So,
Okay, so like my brother has green eyes, but both my parents have brown eyes.
Yeah.
That can happen.
That can happen. So, I'll get to that in just a moment.
Yeah, so also
Wait, hold on.
so mom and dad both have 8, or they have 4?
They have 8, but they're giving 4. They can give any 4 because of chromosome switching and mixing during mitosis. Do you remember how like the chromosomes can wrap around each other? So because of that, you can have any possible combination. So in this case, the reason I made them all positive is because now there's no choice. But what happens if mom has 6 positives and 2 negatives?
Is that like light brown?
That would be like a light to like a medium brown. So she could give all 4 positive, right? But she could give half and half. Now let's say that the father is the same thing. So he could give all positive, or he could give half and half. So if he gives half, and she gives half, What's your kid? They're hazel. And that's what you're talking about, Lakin. So if they have brown eyes, but they could still give 2 each. And now you've got a kid who's like perfectly split down the center, probably have green to hazel eyes.
So how do you get brown to blue?
It's harder, but it can still be done. So let's imagine for a moment that you've got this person who's got this. So the mother gives this exact thing. And then you've got the father who's still brown-eyed, but a little less so. And he gives. So now this person's going to have blue eyes, probably dark blue, but still blue eyes. So what tends to happen in this kind of a genetic pattern is it looks like this. Over time, as you've got more and more combinations, the vast majority of people will end up in this green hazel with a few people on either side. What's this called, by the way, this particular shape? A normal distribution. Now, if you take a look at the possibilities, so how many possible combinations are there here? 4 genes, 8 spots, 2 possibilities each. 16. Okay, so when we had 1 gene with 2 possibilities, how many combinations were there? 3. All positive, all negative, or 1 positive, 1 negative, right?
Right.
So if you remember when we did carrier, we ended up with something that looked like this. So half of them were positive, negative. Half were positive, or a quarter were positive, and then a quarter were negative. When you break it out to 16 possibilities, all you do is you make more possibilities, but this middle part stays just as big. Does that make sense? So you're making a smoother normal curve, but it's still a normal curve. This is just a very blocky normal curve because there's only 3 possibilities. So the more genes that are involved, the more normal distribution it'll appear in the population. Does that make sense? Okay, so what is the key thing to know about polygenic inheritance? Lots of combinations are possible, which results in a more even normal distribution. Height is another example. Now, height isn't controlled by a single gene, but it's controlled by multiple genes interacting with nutrition also, and sleep and rest. But on average, people tend to be average height. But then there's some people who are taller, some people who are shorter. And what happens when you get a short person who marries a tall person? What happens with their kids?
Average.
Some of them are average, but some of them are much taller and some of them are much shorter. What happens when you have 2 average-sized people?
Average.
Most of the kids will probably be average too, but some of them could be much taller, some of them could be much shorter. And then none of that matters if you don't get enough nutrition at the right time of your life. So it's not quite as common now, but about 15 years ago, there were a lot of Guatemalan immigrants in this area, and you would see them walk down the street as a family, and mom and dad are like 5'1", and their kids are like 6'2". Was that just freak genetics? What it probably was is that back where they grew up in the mountains, they didn't quite have enough food to eat at a relatively young age. And as a result, they never grew to their full potential. But their children living here in the United States, eating enough nutrition, were able to grow to their full potential. Does that make sense? So all this genetic stuff is potential, but it interacts with the environment. And that brings us to our next category of genetics, which is called multifactorial. Now, the vast majority of diseases are multifactorial. So there's multiple genes. And not all of the same kind. With eye color, what were they all producing? They were all producing the same thing. What was that thing?
Melanin.
Melanin. In the case of multifactorial, they can be genes that code for different proteins. So multiple genes and interacting with the environment. Does that make sense? Coronary artery disease. So coronary artery disease is a really good example of a multifactorial disease process because you've got inflammation, so you have all of the genes that code for inflammation. You've got high blood pressure as a risk factor, so you have all the genes that code for high blood pressure. You have clotting as part of it, so all of the genes that code for clotting. And then on top of all of that, you have the the environmental interaction of your diet, your exercise, whether you smoke or not. And then even high blood pressure itself. Some people are extremely susceptible to stress for blood pressure. So if they do stress management techniques, they can actually bring their blood pressure back down to normal. Whereas other people, it has nothing to do with their stress, and it just has to do with physiological factors. So you've got a very large number of things interacting together to determine whether a person has coronary artery disease or not. What is that model called? Multifactorial. All right. Any questions about multifactorial? All right, the next thing we need to talk about are a few terms that describe how genetic things are expressed. So the first one is called penetrance. Penetrance is the percent of the population that will have a phenotype given that genotype. And that sounds really weird. So what do we mean by genotype? The genes that code for it. And what do we mean by phenotype? What actually appears. Now, I'm going to give an extreme example, just so you can understand. Do you guys know what intersex is?
Yeah.
Right. So it's a person who is typically genetically male. They have a Y chromosome, but the way that we become boys instead of girls is if you have a Y chromosome, it changes the proto-ovaries into proto-testicles. The proto-testicles produce large amounts of testosterone, and that in turn turns the little developing fetus from a girl to a boy. For whatever reason, sometimes those proto-testicles do not fully form, and so you have a person who's born with kind of like girl genitalia, but seems to be a boy, and genetically they're a boy. And that's typically what we call intersex. Now, a long time ago, and sometimes they'll be born with like a penis but no testicles, because the testicles are still inside, and they're undescended. So when this was occurring like in the 60s and 70s, and doctors like, I don't know what to do. Let's just tell them to cut it off, dress them in dresses, and they can be girls for the rest of their lives. No one has to know. But as it turns out, because their bodies were producing testosterone, they felt like boys. Like most of them never felt like girls no matter what happened. They still felt like boys. So this has come into the news a little bit lately with trans athletes. But there's one guy I'm drawing a blank on his name, from South Africa, and he's actually not a trans athlete, he's an intersex athlete. So he has testes inside of him producing testosterone, but he has no external genitalia that look like a boy. So is he boy, is he girl? So that intersex case, they're genetically male, but what do they look like on the outside? Do they look like a boy?
No.
To some degree, yes. To some degree, no. Sometimes not at all. And that's what we mean by penetrance. Now, so in that case, the genotype didn't penetrate into the phenotype. The phenotype is what they look like. So 99.999% of the time, if you're XY chromosome male, what are you going to be phenotypically? XY chromosome male appearance as well. Now there's genetic variability, there's variability within that, but the idea is 99.9% of the time, what you are genetically in terms of your sex will show up on the outside. So that would be a very high penetrance genotype phenotype. Does that make sense? There are others, I'm sorry, I'm drawing a blank on a good example at the moment, but there are others where maybe it's only 80% or only 50% penetrance. So you have the genotype to have that potential, but it just never shows up. And so one example, actually I just got a good example of it, would be the vulnerable brain theory for schizophrenia. You could have a genotype that is perfect for schizophrenia, but if you don't have that precipitating event while your brain is vulnerable, you will never actually get the disease. Does that make sense? So that would be an example of penetrance where, in that case, you would want a low penetrance, right? Yeah, because the fewer people that have penetrance, the fewer the people have schizophrenia. So there are some cases where we'd want a high penetrance. There's other cases where we might want a low penetrance. Question?
For the intersex, so this is just like a question of curiosity. So can they not have children?
That is a very good question. So if an intersex person has functioning testes on the inside, it is conceivable that they could somehow get sperm from them and then use artificial insemination or in vitro fertilization to have a child.
But they don't. Do they have women? Because women's genitalia on the outside?
It's different for different ones. Yeah.
I'm not looking up.
Yeah. Yeah. It's not. Yeah.
It's like not one or the other.
Right. It's not one or the other.
Okay.
Yeah. Another example. Now, this is not penetrance per se, but this is just an example of interaction with the environment. Is spina bifida. So in spina bifida, you've got normal genes that code for a normal spinal cord. But because you don't have the raw materials that are appropriate, you don't have enough folic acid, you can't fully form the spinal cord and the child ends up with spina bifida. So that's a case where it's congenital, but it's not genetic. The genetics were there to have a normal spinal cord, but you didn't have the right raw materials to make that spinal cord for the child. Does that make sense? Or for the child to make it for themselves. So that'd be it's kind of an example of multifactorial. All right, so the penetrance was one. The other is variable expressivity. So just as an example here, we talked about cystic fibrosis, and before we had modern medical care, most children would die of cystic fibrosis before they were 2 years old. But every now and then there'd be one who lived longer, 6 years old, 9 years old, but very rarely. So that is what we mean by variable expressivity. So some people have it more severe, and then they may have an earlier onset. We talked about that with Huntington's disease as well, that on average a person gets it in their 50s, but I've seen patients who had full-blown Huntington's in their mid-20s. So in that case, they would have more of the protein, which in turn made the disease manifest more strongly and earlier in life. So those are the concepts of variable expressivity. Another one is called a lethal allele. Now, there are certain alleles that, when expressed, have the potential to kill a person. They're called not compatible with life. But most of us only have one of the pair. If you have two of the pair, we think that's one of the major causes of a lot of miscarriages or stillbirths, that the child had a genetic lethal allele, and so that they were never going to survive. Now, what happens when you marry someone who's closely related to you? Or not even marry, just procreate with someone who's very genetically close to you? Say your brother. Your chances that you share the same lethal alleles go up exponentially, which is one of the reasons why when you've got cousins marrying cousins and you have brothers who have children with their sisters, the chances for birth defects goes way, way up. It's because of those presence of lethal alleles.
So what's the actual definition?
A lethal allele is an allele that when you've got both copies of it is not compatible with life, at least not for long. So it's a major cause of birth defects and stillbirths. And they're more likely when you're married to someone who's closely related to you genetically. So there are medical reasons not to marry your cousin. All right. Is there another word on there that on your slide? Oh yeah. So genotype, we already talked about that. What your genes actually are, the coding for, and then the phenotype, what it expresses. All right. The next thing we're going to talk about is epigenetics. The word epi, or the prefix epi, means surrounding or around. And epigenetics is one of the control mechanisms for expressing and controlling genes. So there's two major pathways that we've discovered. And these are things that turn genes on or turn genes off. And we discovered them by looking at like large, population trends. And in particular in Scandinavia. Now, what do you know about Scandinavia? Do you know where Scandinavia is? Do you know what it is? I got a nod. What is it?
Like Norway.
Yeah. So Norway, Finland, Sweden. Sometimes we'll include Denmark. So, now, what do Norway, Finland, and Sweden have in common, especially at the northern ends of those countries?
They used to have Vikings.
Really
Cold weather.
cold. So a lot of times there wouldn't be a lot of food. And when there's not a lot of food, what do?
Starve.
You starve. So when that happens, it kind of genetically selects for people who are better at storing energy and not using a lot. Now, what happens the next year if, say, there's a lot of food? All those people who are really genetically geared towards not burning fat or not burning their energy, when there's lots of food and they eat, what happens to them? They get fatter. So you can have this kind of like thing that happens. Now, what's interesting is that the children who were born to mothers during a time of more food than usual were different. And so were their kids. So something happened in the environment that affected their genes. And it wasn't that their genes were mutating, it's that certain genes were being turned on or off, and then that on or off status was being passed down to the children. Does that make sense? So there really is a case to be made that to be nice to pregnant women because you don't want their kids to be messed up. I said there really is a case to be made to be really nice to pregnant women because you don't want their kids to be messed up.
Oh, like that environment?
Mm-hmm. Yeah. Now, we already know that certain things during pregnancy can directly harm a fetus, such as alcohol or cigarettes. Fetal alcohol syndrome, cigarette babies tend to be smaller and have more respiratory issues, stuff like that. But we're not talking about that. We're talking about their genetics are turned on or off in such a way that can then be inherited later on as well. And that is called epigenetics. So there are two types of epigenetic phenomena we need to know about. The first one is called methylation. Now, what do you think that means?
Are those the drug addicts?
Methyl group.
It has nothing to do with meth addicts.
It's a methyl group.
It is a methyl group. For those of you who had principles of chemistry, with people who shall remain nameless, what is a methyl group? I don't know. Okay, so I'll remind you. It's a carbon with 3 hydrogens surrounding it. So that's a methyl group. Now, when you attach a methyl group to DNA, it kind of like ties it up and holds it in place. So methylation of a chromosome or of a gene turns that gene off. It's like a light switch, on or off. So to turn that gene back on, what would you need to do?
Remove the
Demethylate it. So you have to get rid of the methyl group.
Is that the other one?
No,
There were two. Oh, okay.
no. To methylate, you tie it up. And then you take the methyl group off, you untie it.
So it's off if it's tied to.
It's off if it's methylated. So methylation's like tying it up, turning it off. Now, the other possibility is the production of what are called histones. Now, histones are kind of like a knob. The more tightly you wind the DNA around it, the less it's expressed. The more loosely you tie the DNA around it, or wrap it around it, the more it's expressed. So instead of being an on-off switch, it's more like a dimmer. So methylation, on-off. Histone, you can turn up or down the what?
Dimmer.
The expressivity. Like a dimmer.
So the tighter it is, the less it's expressed, the easier it is.
Yes. And I've given you like a 3rd grade or maybe a 5th grade level YouTube video that you can watch about epigenetics from SciShow.
Yeah.
So if you haven't watched it yet, you can watch it. I think it's pretty funny. What's funny is that people will watch the whole video and then go, whatever. But these things will show up on your test. So don't just, oh, that was funny. That guy Hank, he's funny. Yeah, you actually need to understand what he's saying. And remember, it's being aimed at 5th graders. So I expect that you college kids can handle it. That is like a really bad expectation. And yet it's what it is. Okay, so the last thing that I'm going to mention about control mechanisms is that epigenetics is just the latest hot topic, but there are a number of other control mechanisms that are in place in your DNA. So there's what's called microRNAs, which is an RNA that doesn't make proteins. It's an RNA that suppresses genes, turns them off. So that's one example. We also have DNA control mechanisms. Repair mechanisms. So one of the major problems in today's society is cancer. And what causes cancer is genetic mutations. And our bodies have an enormous amount of our DNA is geared towards repairing DNA. So it's DNA that codes for repairing itself, if that makes sense. So that's another thing that we have to think about. And then the last thing I want to mention is just the genetic testing. So we previously talked about just because you can get something tested doesn't mean you should. And you should think about what the ramifications of that knowledge are. And now I'm going to give you an example that has nothing to do with disease. Instead, it's going to have to do with athletic performance. So there are certain genes that are more highly associated with building muscle and an increase in muscular performance. Now, if you as an athlete get tested and you find out you don't have those genes, what does it do to you? It lowers your confidence and it lowers your expectation of performance. What does that do to your actual performance? It lowers it. And this is what's called a nocebo effect. Now, how many of you like to watch the Olympics? Personally, I do not. The Olympic Committee makes billions of dollars off the Olympics, and the athletes basically get nothing. If they're lucky, they get a Kellogg's Fruit Loops commercial out of it. But most of the athletes are, they go poor trying to compete. So I don't think that's very ethical. So I don't watch the Olympics anymore. But the important thing is, how many of you have seen, you watch the Olympics, nowadays they have all that tape all over them. Have you ever noticed they've got like tape on their shoulders and tape on their hip and tape on their knee? And The evidence that that does anything is basically zero. Or Michael Phelps and getting the cupping, the evidence for that is basically zero. But if it makes them feel like they can do better, guess what? They actually do better. So the opposite of nocebo, which is where you expect something bad to happen and it does, is placebo. So in that athletic context, knowing that you are missing a certain gene for athletic performance makes your athletic performance worse than it would have been otherwise. So why would you want to get tested when it won't help you, but it could hurt you? Well, nocebo is when you expect something bad to happen and it does. So like let's say that you're going to lift some weights. You're like, oh, this is going to be heavy. This is going to be so heavy. And then it feels heavy. Well, you made it feel heavy. The opposite of that might be called the magic feather effect. So how many of you seen Dumbo? What does Dumbo need to fly? The magic feather. But as it turns out, it's not a magic feather at all. It just gave him confidence. What would that be? Placebo. Okay. So it's those terms have very specific meanings in the context of medicine, but we also use them not in medical context, in terms of medicine context, to mean some of these behavioral things. So it's called the biobehavioral model of health. So it's not that our bodies are just machines that are mindless, but our mind interacts with the machine to produce results. So when you tell your mind, you have this disease, you have this problem, it becomes a self-fulfilling prophecy to where you actually end up manifesting that. So, and when we go back to penetrance and variable expressivity, you could have a gene for a genetic disorder, and yet you live till you're 70 or 80 with no symptoms or sign of the disease. Meanwhile, the person next to you has that same gene. Maybe they're your brother. They have the same gene and they get the disease at age 40. And when you get tested at age 20, what are you doing to yourself? You're psyching yourself out. You're making it potentially more inevitable that you get the disease. Because stress and worrying about things can actually hasten the diseases. We're going to talk about cancer later in the course, but there was, you guys know what 3 Mile Island is? No? So in New York, there was a nuclear power plant on a place called 3 Mile Island, and it melted down in I think it was late 70s. And there was radiation that leaked out. And cancer rates went up surrounding 3 Mile Island afterwards. But the cancer rate increase had nothing to do with the actual exposure to radiation. So what the researchers concluded was it was worrying about radiation is what was causing the cancer. So the whole point of it is genetics are destiny to some degree, but what you think about your genetics also matters.
So if you worry that you're going to have cancer, you can get cancer.
Potentially. That's what they concluded in that particular study. I believe that that's what they concluded in that particular study. So is it a possibility? Yeah, I think so. I think it's definitely enough to where we shouldn't think that just because I have this test result means I'm going to go this way. Another example is testosterone levels. In male athletes, a lot of the highest performers actually have relatively low testosterone. But if they get their testosterone checked, I've got low testosterone, oh my gosh! And next thing you know, their performance goes down.
So should you not get tested for the BRCA gene, or is that something you should do?
So, should you get tested for the gene or not? If it's going to change what you do, like say you already have symptoms, or there's a treatment that can prevent it before symptoms occur, then it's probably a good idea to go ahead and get treated. But if there's...
Like for breast cancer, if you get a
Yeah.
mastectomy before even having breast cancer,
Right.
a good idea.
But the problem there is that double mastectomies are associated with something we call cytokine storm. So remember cytokines from inflammation. And that cytokine storm, the inflammation itself can be pro-cancer. So we're not sure. And, there's one of those things where it's like, to some degree, when it's your time, you can't escape it. So this was a patient that I actually knew. I didn't treat them, but one of my colleagues treated them back when I was still working as a nurse practitioner. Her mother and her sister had both died of breast cancer. She was positive for the genes. So she decided she was going to get double mastectomy. And then she died about 2 years later from ovarian cancer. So it's like, was it her double mastectomy that caused the ovarian cancer? No one knows. Could it have played a part in making it worse? Possibly. The answer is we just don't know.
Because of the
I don't even know it's 50-50. I mean, maybe it's 25-75. I don't know. But if the treatment isn't benign, then worrying about it excessively and the things we do about it once you know could be worse. So just example, just getting tested. Nowadays the recommendation is that women no longer do self-breast exams because 90% or 95% of the lumps that were being found were non-cancerous. But she was getting all of this anxiety. Then she was going and getting more testing done. Then she would have to go and get a biopsy done. She's getting more risk for health exposure to x-rays. She's also getting higher health risk because of the exposure to risk for infection, the surgical procedure. If there was actually a cancer there, by poking a needle into it, now you're creating a needle track, which is called seeding, and so that can actually cause the spreading of the cancer. So it's like all of these things that we're discovering, the things that we thought made sense, sometimes don't make sense. So, but the key thing is, if you don't already have symptoms, and there's no effective treatment for what you're gonna do anyway, probably better off just not getting tested. And I'll actually link to I think it's like an hour and a half long talk by two physicians who are talking about this issue of getting tested, somewhat in the disease context, but also within the context of sports performance. So you can watch that if you're interested in the subject. They've got further reading you can do there as well. All right, so that is going to be it for genetics for right now. Any questions? All right, let's take a break. All right, welcome back from our break. Like I have to talk like that for the video. Hi, Mom. Okay. All right, so can't do it.
don't want to anymore. Don't look at us.
Who are you?
Well, we're not doing diabetes.
I can't believe you guys watched that, read that decapitated chicken story. You guys are a bunch of sickos.
You told us to.
I'm sending you that YouTube video right
It's hilarious. He just has no head.
Just running around. I totally asked my mom about it too. I was like, "Do you know about this chicken thing?" Before I watched it, and they were like, "No, what are you talking about?"
Nope.
Oh
Yeah.
God.
But yeah, if you cut the head off of a chicken, its body will still run.
Yeah.
For years and years of this chicken.
Yeah, that's weird. They must have left the brainstem intact.
Yeah, that's nothing. They had a blood clot.
Oh, remember the Band-Aids that
You did not watch it?
I didn't finish it.
All right, back to our story.
I haven't finished it. I'm saving it for later.
All right, back to our story. So what we're going to talk about now is HIV/AIDS. So in the coming plague, for those of you that actually read the book, there is a chapter on HIV. It's, I think, the longest chapter in the book. And it's the plague that has stayed with us in the modern Western world. Most of these kind of, I say plague, most of these infections that it talks about in the coming plague are tropical diseases, things that are out in exotic places. Maybe there's a couple cases in the United States or Western Europe, but for the most part, they're in these kind of isolated rural areas, often in Africa or Asia. But HIV has spread throughout the entire world and is endemic in large portions of Africa and Asia, but it's also endemic to smaller extent in Western Europe and in the United States. So it's the unique one of those diseases in that regard. Now, the way that we first found out about HIV was its pattern of who it was infecting. And because it was infecting a very small number of people, that led us to certain directions politically. And so it started off showing up in drug users and in patients who were men having sex with other men. So in its first iteration, we actually called it GRID, which was gay-related immunodeficiency disease. And then it eventually became autoimmune deficiency syndrome, and then eventually became what we call it now, acquired immunodeficiency syndrome. It's also present in Africa, and the theory is that somehow it started in Africa and then, through tourism primarily, was transmitted first to Haiti, and then through sex tourism and drugs came to Miami and New York, and then from there kind of spread out the rest of the country. But we're not really 100% sure where it came from, but that's the kind of the theory that we are operating under. And in Africa, AIDS looks very different than it does in the United States. So one of the things we'll talk about briefly toward the end is some of the differences in the disease pattern and what could possibly account for that. But the theory at the moment is that the virus, HIV, which is human adenovirus, infects our bodies, targets CD4-bearing cells, which is primarily going to be which kind of cells? The helper T cells. And then from there, a person will have fewer and fewer T helper cells until eventually they can no longer mount adequate immune defenses. And then the person ends up with what we call AIDS, and then dies of an infection or of a rare cancer. So remember, our immune system, in addition to fighting infection, also helps to destroy cancers. So when you don't have a well-functioning immune system, you're more likely to die of cancer. When AIDS was first discovered, it was considered a death sentence in that you were probably going to die within the next 1 to 2 years. But we now have people who have lived with HIV that are documented for 30 years or more. So like kind of the big one that we think about is Magic Johnson. He was a basketball player. He was infected, I want to say in 1991, but it may have been in 1992 that we were aware of. And so here we are now in 2019. How many years later is that, for those of you good at math?
26.
Say again?
26.
26?
92 would be 26.
Oh, I was going to say 91, but okay.
91.
Yeah. So it's about 28 years, is what I was thinking. Yeah, it's good enough. Close enough. And that was when he was diagnosed. So he probably caught it earlier than that. So let's just round it up to 30 years. So here's a documented case of someone who's lived with HIV for almost 30 years and has no sign of AIDS. So the important thing for us to know is that, first of all, infection with the virus does not equal AIDS. What is the idea? That HIV leads to AIDS. So what is AIDS then? It's typically defined as a CD4 count of less than 200 and a disease. Now, which disease, you ask? Well, one of the AIDS list. Now, do you think that definition is a little loose? What kind of disease? Well, one on the list. So the list includes some pretty vague things. Some of them are very specific. So like in the early days of AIDS, a lot of patients would die from Kaposi's sarcoma, which is a very rare skin cancer. So that's one of the things on the list. But there's also some relatively mundane things on the list as well, like pneumonia. So one of the things that's also difficult is if you look at some of the statistics in Africa, they'll say, well, in the country of Zimbabwe, 50% of the population has HIV. Now, first of all, how do they know that? Do they test the whole population? As it turns out, when you look at the studies that claim that, they're basically saying anyone who had any of these infections, we're going to consider that they've got it. So they're not even necessarily doing blood testing to come up with some of those numbers. So why would they do that, you ask? Well, the cynical side of me says funding. Because if you rely on governments to give you money, what do you want to do? The problem is as bad as possible. Now, that's the cynical side of me. I don't know. That's just what my cynical. Don't be cynical. Be good. All right, back to our story. So in Africa, sometimes they actually skip this part and they skip that part and they just look, do you have a disease on the list? And remember, some of those diseases can be rather mundane. So when you read a study or you hear on the news about HIV and AIDS, one of the things you need to do is look at how they actually studied it to know how good that reporting is. Does that make sense? Okay. With modern treatment for HIV, we have patients who never progress from HIV to AIDS. Now, so let's talk a little bit about HIV first, and then we'll finish up talking about AIDS again at the end. So, HIV, the H stands for human, and we believe that it can only be passed from human to human, can't go to any animal reservoir, can't be transmitted by mosquitoes, things like that. It's a rather fragile virus, and it's transmitted primarily through sexual contact. It can also be transmitted by blood contact, which is why it's fairly common among injecting drug users, because they share needles and reuse needles a lot. It also is one of the reasons why it shows up in men who have sex with other men more so than in heterosexual populations here in the United States. Now, that's another difference between Africa and the United States. In the United States, if you look at the CDC numbers, the vast majority of cases of HIV are men who have sex with other men. In Africa, the pattern there seems to be much more heterosexual, and it's women who tend to get it more than men. Now, one of the potential explanations for that is some of the wars that have occurred in Africa. And so you have families being dispossessed, and you have women who are following soldiers and basically acting as prostitutes. So they're acting as that magnifying agent. Because they're having sex with multiple partners, they're more likely to get HIV. Whereas in the United States, because it's the men who have sex with other men, they tend to have more sexual partners. Even when they're in a monogamous relationship, they will often have sex with multiple partners. So it's like, yes, I live with this person who's my husband, but I can see other people on the side if I want to. If you read the book, The Coming Plague, the CDC in the late 70s, early 80s, at first they were defining promiscuous among homosexual men as 10 partners or more in a year. But it turns out that some of the people they studied were having 500 partners or more in a year. So if that kind of would explain some of the magnification of the disease spreading in that small population. Now, that's not to say that every person who has sex with other men does that, but enough of them do that it was able to kind of amplify the epidemic in that population. So even today in the United States, still the vast majority of people who get HIV is from sex with other men. Now, men with sex with other men, MSM. Now, why don't we say homosexual or gay? Because not all of them identify as either homosexual or gay. So a lot of them they consider themselves heterosexual. They may have a wife and kids, but are still having sex on the side with other men. The pattern is there that in the United States still has primarily men who have sex with other men. Now, what are we being taught in high school? Use protection. And who are we being taught can get HIV? Everyone and anyone. Now, to some degree, that's true, but it's not 100% true. And if you look at the CDC figures, you'll see that women who get it are quite low, and men who are heterosexual only are quite low. So why are we told that anyone can get it? And again, the cynical side of me says, not a scare tactic, but when you rely on taxes for funding, well, if we can get everyone to be concerned, then they're more likely to fund it. But if it's them and their problem, well, then maybe we're not so willing to fund it. So I don't know if that's the reason, but I suspect that may have part of the reason, be part of it. But it can occur in all of the different age groups. However, there's just, in Africa, more likely to be heterosexual women. In the United States, more likely to be men who have sex with other men. And that's partly why, when you go to give blood, they ask you, like, have you had sex with another man? Have you paid to have sex with another man? Have you ever paid to have sex? And you're like, what? I don't even understand the combinations of what you're asking now. Have you ever lived in Africa? Have you ever been to Africa? Have you ever done sex tourism in Africa? Have you ever, and you're like, why do they ask you in so many different ways? Because what people do is they rationalize. No, I've never done it that way. Yeah, like, have you ever been alone in the room with the other person? No, because I was there too, so I couldn't have been alone. Yeah, which is partly why, how many of you have ever flown in an airplane? What does it say on the smoke detector in the bathroom? Do not tamper, destroy, mess with, like 6 different words. Well, I didn't destroy it. I just tampered with it. Yeah, so if you ever wonder why nursing handbooks are so big, is problem nursing students. But it didn't say that. Okay, well, it's got to say that from now on. So that's also why syllabi tend to get bigger over time. So the bottom line is, it is primarily transmitted through sexual contact. It is present in very small amounts in saliva, but we don't think that it can be transmitted by saliva. Pretty much the chances are just very small. Doesn't live on toilets or anything like that. So the primary thing is sexual contact, blood contact. Once it gets inside the body, what it's going to do is it's going to infect CD4 bearing cells, which include your macrophages, some macrophages, some dendritic cells, but primarily CD4 cells. And so we need to know the life cycle of HIV. So here we've got a T cell, and here's its CD4 on the outside, and our little virus has a receptor that can bind to CD4. And then it's invited inside. So the very first thing that our virus has to do is to fuse with the cell. There are some enzymes that are associated with that. And the reason why I say this is that we have drugs that can inhibit that fusion step. Does that make sense?
It has to bind with
It has to bind with the CD4. So the reason it infects CD4 is because it has proteins that can bind to CD4. Okay. And then the cell says, oh, come on in. So the first step, it has to bind or fuse with the CD4 cell. That's an important step because we have certain drugs that can inhibit the fusion. And what do you think we call those drugs? Fusion inhibitors. You're so smart. Okay, step number 2. These viruses are called retroviruses. And the reason they're called retroviruses is not because they like 60s music and bell bottoms. You can tell they're out of style because today it's all skinny fit. The reason they're retro is because they don't have DNA of their own. What do they have? They have RNA. So this little guy's got RNA inside it. And what it has to do is it has to take its RNA and put it into DNA. So it has to somehow convince the T cell to take its messenger RNA, turn it into DNA, and then make it part of the genome. So now the cell thinks that this newly created viral DNA is part of its own DNA. So that is reverse transcriptase. So what is transcription? What's transcription? We talked about this just a few minutes ago at the beginning of genetics. Well, that explains it. I don't remember nothing from genetics, especially not the intro. Okay, so we take DNA and we make a copy messenger RNA. Now we're doing that backwards. We're taking viral RNA and we're going to DNA. So that's reversing the transcription process. The enzyme responsible for that is called, read it on the board. Reverse transcriptase. It is amazing to me how many of you guys cannot read what's on the board. And I know my handwriting isn't great, but it's not that bad. And I haven't even filled up the board yet. What can you do? So we're taking viral RNA and we're turning it into what? DNA. Now, our next step is we have to take that DNA and we have to integrate it into the cell's DNA. And what do you think the enzyme that does that is called? Integrase. So integrase integrates the viral DNA into the cell's DNA. And guess what we have? Oh, we have reverse transcriptase inhibitors, 2 different flavors, and then we also have integrase inhibitors. It's fun, huh?
Okay, you're spelling it throwing RNA.
I-N. That's how you spell it. Oh, yes. Integrase. Integrase. It's touch and go there.
What's the difference between the, like, in the PowerPoint it said ssDNA and dsDNA?
Yeah.
Say it again.
In the PowerPoint it says ssDNA and dsDNA.
Single strand and double strand. So when you take the messenger RNA, or sorry, the viral DNA, and you turn it into DNA, it'll be a single strand. So then you've got to take that single strand and give it a double. Yeah. 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 Yeah, I'm assuming that this is in there. Okay. So then the next step is we're going to make viruses, or make the viral proteins. And who's in charge of making the viral proteins? The DNA doesn't make proteins. It codes for proteins. What makes the proteins? Well, they're made in the ribosome, but what is responsible for making it? The cell. Which cell is it? Well, it is a eukaryotic cell, as are all of the cells in your body other than red blood cells and platelets. Yeah? Go ahead. Okay, the CD4 cell, so the T helper cell. So the CD4 cell is going to make viral proteins using the viral DNA that was integrated into it. And then those viral proteins are going to go towards the edge, towards the membrane of the cell. So we've got our little viral proteins assembling over here. And then basically the cell is going to kind of pinch off like this with the virus inside, and then you've got a new little virus. So what is that called when it kind of pinches off and you birth a new virus? What do we call that?
Isn't it like something with like a moth?
It's like a tree in the springtime. What's a tree? Close.
Oh.
Flowers bloom and trees bud.
Yeah. Okay, like nothing like cleavage.
Yeah, that's what I was thinking.
So viral budding. So we assemble the virus in step 5. Now, during the viral budding, the very last step occurs, and that is when we cleave the proteins into active viral proteins. So remember we talked about how proinsulin gets made and then you have to cut it to become insulin? Well, those viral proteins are not active until they're cut. And at that moment is when proteases, which is a fancy way of saying protein cutters, cut the viral proteins. And that's important because we have drugs that can inhibit that. What do you think we call them? Protease inhibitors. So we've got 4 places where we can attack this virus. The first one is fusion inhibitors. Second one, reverse transcriptase inhibitors. Third one, integrase inhibitors. And fourth one, protease inhibitors. Now, we're not going to talk about the drugs just yet, because we're going to talk about what happens to a person when they are exposed. So, when a person is exposed to HIV in a large enough quantity that they actually become infected, there is a window of time where the person can't be diagnosed. That window has gotten smaller and smaller. So, back in the 80s, we had to wait 6 months before we knew whether you actually had HIV or not. So if you got a needle stick and the person that you got the needle stick from had HIV, you had to wait 6 months before you could be sure that you were not going to be HIV positive. That window now is much, much, much smaller. You can sometimes detect HIV within a couple days, but generally you need to wait up to 2 weeks to be definitive. And we typically will do a 2-step process. So the first step in the process is a very sensitive test. What does sensitive mean? It can pick up almost all the cases. Now, what is the problem with sensitive things? They could be wrong. What do we call that? False positive. So, if a person is positive in the sensitive test, then we're going to do a second confirming test that is more specific. What does specific mean? Fewer false positives. And if both of those tests are positive, chances are really strong that you've got it. If the second test is negative, then it's much more likely that it was a false positive. Does that make sense? The second confirming test is almost always a Western blot.
Is the first one the ELISA?
The first one used to be ELISA. Nowadays, most of the time, it's a rapid. But they call it ELISA, not ELISA. So these tests are techniques, and they're not just used for HIV. They're used for other things as well. One of the things you could use an ELISA test or a Western blot for is detecting HIV antibodies. But it's not the only thing we can use them for. So the rapid ones are typically you put it in the patient's mouth and you swab the inside of their gum. And then it basically is like a pregnancy test where you put the little thing in the thing and you pop the thing and then you wait 15 minutes and if it turns blue, that's bad. And if it doesn't turn, well then chances are you're okay for now. Does that make sense? Okay. Once a person has a diagnosis of HIV, there's 2 further tests that we typically would want to do. The first one is called a viral load. And what we're doing is we're looking at the number of viruses in a given amount of blood. The higher the viral load, the more severe the infection, and the more likely they are to progress to AIDS. The lower the viral load, the less likely they are to progress to AIDS, or the longer it'll take to do it. So when we give drug therapy, what is our target? Lower the viral load. The second thing that should be done for this patient, the second test, is a genotype. So in the old days, we just chose some drugs and gave them to you. Let's see if these work. The problem is that HIV is very mutagenic. So it really easily mutates. And there's quite a few strains of HIV that are resistant to multiple drugs. So there's no point in giving you a drug that that the virus inside you might already be resistant to. Does that make sense? So what we're going to do before we start therapy is we're going to get a genotype. And that way we'll know what would be the better drugs, not necessarily for you, but for the virus inside you. Does that make sense? It might still be a bad drug for you in terms of adverse effects, because that's your genetics and your cytochrome P450. But in terms of will it be able to prevent that virus from growing, that's what the genotype tells us. Does that make sense? Okay. And then when we start a patient on drugs, we want to use what's called HAART, H-A-A-R-T. Highly active antiretroviral therapy. Highly active antiretroviral therapy. So, what kind of virus is HIV? A retrovirus. When we fight retroviruses, what kind of therapy are we giving?
Highly active.
Antiretroviral therapy. And the highly active means you're going to give at least 2 different drugs that work in different pathways. And oftentimes you'll use more than 2.
2 different drugs that work in one pathway?
2 different drugs that work by different pathways. So you wouldn't give 2 fusion inhibitors. You would give a fusion inhibitor plus a reverse transcriptase inhibitor. You might give 2 reverse transcriptase inhibitors plus a protease inhibitor. But you always have to have at least 2. Now, when the first antiretrovirals came out, they were not very good drugs in the sense that they had a lot of side effects. You had to take them sometimes 4 to 8 times a day, and they were just kind of nasty. They're super expensive. Well, they still are expensive, but there's lots of programs to help patients afford them. But they're expensive, and they have lots of adverse effects, and you had to take them quite often. And a lot of patients would just go off the drugs because the side effects were just too much for them to handle. The drugs nowadays are much more tolerable, and we have much better formulations that you don't necessarily have to take 4 to 6 times a day. So how many of you have ever taken an antibiotic you had to take 4 times a day? And how many of you took it exactly as you're supposed to for 10 days? How many of you messed up after 3 days? So now you've got a drug you've got to take for the rest of your life. That way you can see why it would be better to have a drug you only have to take once or twice a day instead of 6 to 8 times a day, right? Okay. HIV drugs, or HIV in and of itself is a subspecialty. So we're not really going to go into a lot of the drugs. We're just going to do an overview of the drug classes, and then we'll talk about one drug that is used as prophylaxis. So if you get a needle stick, there's a drug you can take that will help reduce the risk of getting the HIV transmission. If a mother is about to give birth to a child and the mother is HIV positive, giving the mother the drug before labor will help prevent her from passing it to her child during labor and delivery. And if you happen to be married to a person who has HIV, then by taking the drug, you can reduce the risk that you will contract HIV from your partner. So that's what we use that for. All right, so at this point, are there any questions before we move on to drug classes? HAART. What does it mean to do HAART? At least 2 different drugs from 2 different classes.
And you use that for those 3 that you just said? You use a HAART? Or that's just the overall concept of how you're going to...
That's just the overall concept of HAART. Okay, now, in practice, the very first drugs that were discovered, or invented, depending on your perspective, were the reverse transcriptase inhibitors. And reverse transcriptase inhibitors are considered the backbone of HAART. So you're almost always going to see a patient on at least 1 reverse transcriptase inhibitor, sometimes 2, and then other drugs as indicated by the genotype. So, but we're going to start at number 1, just because it's the top. So fusion inhibitors, how do they work? Inhibiting the fusion of what?
CD4 with the virus.
The virus to the CD4 receptor. Okay. So then, reverse transcriptase inhibitors, how do they work? Well, it depends. There's actually two kinds. So we call them nucleoside reverse transcriptase inhibitors and non-nucleoside reverse transcriptase inhibitors. So I know what you're thinking. What's a nucleoside? Right? Or you're thinking, who are those people? What are they doing in here? Okay. So what is a nucleoside? Go ahead. Close. So those are called base pairs, but when you connect it to its ribonucleic backbone, that is a nucleoside. So the base pair, so let's say the adenine plus the ribose backbone, whatever is in here, this is a nucleoside. So a nucleoside reverse transcriptase inhibitor, instead of being a functional nucleoside with adenine or uracil or cytosine, instead it's got drug there. So what this nucleoside reverse transcriptase inhibitor does is it pretends to be a nucleoside, and when you do the reverse transcriptase, you end up getting faulty DNA. Because it goes, it's matching them up, and then it goes, what is this? D? We don't know no D around here. We only know A, U, C, and G. There's no D. Who the heck brought a D in here? I can't work under these conditions. So it's a crotchety, ribosomes are crotchety old men in my, no. But that's the idea with the reverse transcriptase inhibitors that are nucleosides. They pretend to be a nucleoside, and they just mess up the DNA reverse transcripting. Now the non-nucleoside reverse transcriptase inhibitors just bind the enzyme, reverse transcriptase, and prevent it from working. Do you need to know names? You need to know one name. But hold on, we'll get there. We're going to go through all the classes first, then we'll do the one name you need to know. All right. So, any questions about the difference between nucleoside reverse transcriptase inhibitors and non-nucleoside reverse transcriptase inhibitors? You should know the adverse effects, but I'm not going to talk about them in class. So, yeah, for the exam, you might need to know them, although you might not.
So the non-nucleosides, they bind to what?
The non-nucleoside reverse transcriptase inhibitors bind to the reverse transcriptase enzyme.
So they both affect step 1 and 2 distributed.
They both do step 2. Step 2. But they do it in different ways. Now, do you guys remember an antibiotic that pretends to be an amino acid that messes up protein synthesis in the same way? You do? So you're just saying that. But it's aminoglycosides. So the nucleoside reverse transcriptase inhibitors are kind of like aminoglycosides, but for DNA instead of protein synthesis. Yeah, that doesn't help. Oh, well, I tried. Okay, next. The integrase inhibitors, what do? They inhibit the integration of the newly formed viral DNA into the human genome. Okay. So that's integrase inhibitors. And then the protease inhibitors inhibit that final activation of the viral proteins. At one point, so the reverse transcriptase inhibitors were discovered first. First the nucleoside, then the nons. And then the protease inhibitors were discovered. And the protease inhibitors were considered to be the wonder drug. Except, very, very quickly, the viruses became resistant against them. So that's when we decided that we couldn't just give protease inhibitors by themselves because they worked super awesome, but only for about 6 months to a year. And then after that, they stopped working. But by giving them in combination with the reverse transcriptase inhibitors, it made them last a lot longer. And so that's how HEART came about. So I'm an old whippersnapper, I remember. No, I don't really remember anything. So that's where the protease inhibitors are. Any questions about the drug classes? Now, the only drug you need to know, drug name you need to know for your test, is the reverse transcriptase inhibitor that we use as prophylaxis and as a first-line treatment. So that drug name is on your PowerPoints, and what is it? What?
ACT?
You still have AZT on your notes?
Yeah.
Oh, how gauche. Well, that's an old one.
ZDV?
Yeah, that's the same thing.
Tenofovir?
Tenofovir.
Is that the one you want? Okay.
Oh, Tenofovir.
Yeah.
So that's largely
TEN.
Oh, yeah. So it does say the new drug on there. Okay, thank you.
But it's,
Yeah. All right.
That's like a little bullet. It's like a little.
Okay, so AZT was the original one. That was the first one that was really effective. But no one uses it anymore. Nowadays, you're going to see tenofovir. So tenofovir is basically the new AZT. It's a reverse transcriptase, a nucleoside reverse transcriptase inhibitor that is kind of like the backbone of most highly active retroviral therapy. There are others, but that's probably the most common one.
Used for prophylaxis and what else?
And it's also used for prophylaxis in those three populations we talked about. What are the three populations? Needle sticks,
Sexual partners,
sexual partners, and
pregnant mothers.
vertical transmission during delivery. What do you mean by vertical transmission?
Where they come
From mother to child.
Can I just make sure I have this right?
Mm-hmm.
So the four that we just went over, NRTIs, NNRTIs, protease inhibitors, and integrase inhibitors are the ones that are used for HEART.
All of them are used for heart,
Even fusion inhibitors?
even fusion inhibitors.
Okay.
Does it have the same reverse effects as AZT?
Very similar.
Do you want us to know the adverse effects or just
doesn't
the
even have
effect?
adverse effects on here.
If you want to know the adverse effects, what I would recommend is reading the book and or,
Oh, he said.
no, Use Central. Oh yeah. Yeah, I don't know how to log into Use Central. Yeah.
I got it.
What was CCR5 blocker?
Okay, yeah, so CCR5 blockers. CCR5 is a receptor, it's a protein that is part of the process of fusion. And some HIV has it and some doesn't, which is partly why the genotype is important. So in the ones that have it, it can be really effective at helping to prevent infection or reduce the viral load in those patients. But that's one of the more newer drug classes. And at the time we did these PowerPoints, it's just not worth putting them in as something you had to know for your test. And guess what? It's not going to be on your test. So again, HIV is a really complicated topic. We are like barely scratching the surface of HIV and AIDS. So, and like the amount of research that's happening in HIV and AIDS is amazing. And so just from 30 years ago, it was a death sentence. You're going to die within 1 to 2 years, to now you can live 30 years and never have symptoms is pretty amazing. And going from you've got to take drugs 8 times a day, 4 or 6 times a day to, you take a drug once a day. And the adverse effects went from horrific to not too bad. It's pretty medical miracle-ish to me. So, what else do you want to know about HIV and AIDS?
What else is going to be on?
What else is going to be on the test? So we talked about some of the differences between Africa and the United States. So those are important to know. Now, what can account for some of the differences? We mentioned in terms of the transmission pattern that one of the differences is the presence of war in Africa, and that's a potential reason why it tends to be more common in heterosexual women, is that the prostitution that goes along with war.
poverty.
Another one is poverty in terms of especially not enough, or not adequate nutrition, and also not enough clean drinking water.
So people will.
So if you get an infection and you don't have enough nutrition, that's going to impair your ability to mount an infection. What happens to someone when they get a fever? They're using more energy than usual, right? Well, what if you don't have much energy? What if you don't have much reserve in the first place? Yeah, so that's going to begin to destroy normal, healthy tissue. So it's one of the reasons why in Africa, fat people have traditionally been seen as rich and healthy, as opposed to in the United States, where we think that fat people are, lazy and unhealthy. So for most of our world, being a little bit overweight was considered being good and a sign of wealth and health. And it's only in the last, 20, 30 years that we're like, fat, ew, gross, who would want to be fat? Go figure. So in terms of the different patterns that we see among HIV in the United States and Africa, one of them is war and prostitution. Another is inadequate nutrition, and then also inadequate clean or sanitary water. And then you also have the increased risk of other diseases that are around that can be comorbid. So we don't really have that much in the way of malaria here in the United States, whereas you've got a lot more of those types of diseases, and then also parasites in Africa. If you really want a horror story, look up bilharzia or schistosomiasis. But anyway.
For the clean drinking water, why is that affecting it? Like, is it like a cholera thing? Like, where if you like...
Well, not specifically cholera, but yes, infectious diseases.
Could be. So yeah. HIV could be contaminated if someone like used the bathroom in a water thing and then someone drank from it, they could get HIV from that?
No. No, but if they're infected with HIV and they're getting this continuous flow of new infectious diseases from their water, it's going to impair their body's ability to maintain a healthy immune system. And then if you've got HIV, which is living inside of your CD4 cells, you're not able to mount an adequate response. So the idea is that it accelerates the cycle. So what might take a year or 5 years in an otherwise healthy person would take 6 months in a person who has these recurrent infections that are being driven by unhealthy water.
What does it mean when it's undetectable? Does that mean the viral load hits below a certain point?
Right. So when
be undetectable.
Right. When viral load is undetectable, that means we don't see any. So does that mean you're cured?
No.
Now, what happens if you stop taking your drugs and it stays at 0 or undetectable for the rest of your life? Are you cured?
Maybe. I don't know.
For all intents and purposes, right? Yeah. But are you willing to take that chance? Because if you go off your drugs, what might happen? It could come back, and it might be resistant next time. So when it goes undetectable, you're kind of cured for all practical purposes, but we still want you to stay on the drugs.
Can you still spread it, right?
Say again?
You can still spread it.
Can you still spread it? Well, your chances of spreading it are dramatically lower than if you had a high viral load. Is it possible? Theoretically, yes, but generally speaking, the lower it is, the lower your chances of spreading it.
So it's just not considered curative yet because there's not
right.
one thing that will just wipe it all out.
Well, we don't have enough evidence to say that you are cured, because to be cured means we have to withdraw treatment and see if it recurs or not. And
no one's willing to do that.
I wouldn't say no one, but there's no clinical trials. That clinical trial will never be cleared by an ethics board because of the risk of, well, if it does come back, it might not be treatable the next time.
Yeah.
Well, again, what does cured mean?
It's been gone there now.
Yeah, right. But the thing is, there might be a few documented cases where viral load has been sustained at 0, but we don't call it cured at this time. Again, for it to be called cured, there'd have to be clinical trials that would be done where they systematically take you off drugs and see what happens. And so although there might be cases that look promising, we can't say for certain whether it is or isn't at this time. I'm sorry I'm giving you such horrible answers, but that's how it is. Sometimes we just want it to be black or white. Like the Queen song. You don't know what I'm talking about, do you? All right, go home and listen to Queen Bicycle.
We were all just kind of